Here is Jiankui He from China. He's going to tell us about gene editing in humans and monkeys and mouse embryos.
Okay. All right. So my name is Jiankui He. You call me JK. It's easier. I'm from Shenzhen, China. So today I'm talking about the germ genome editing. In particular, I'm interested in evaluating the safety, which is off-targeting and the mosaicism. So a little bit of background of myself. I studied CRISPR evolution when I was a PhD student at Rice University in 2009. And then I moved to Stanford postdoc to natural sequencing, to biomimetics. And then I go back to China, start my own lab. We developed single molecule sequencer stuff. So the question I'm asking in my project is, it's off-target is a problem or it's not a problem. Some people say it's okay, but some people say it's big issues. So I want to systematically evaluate it, in particular for germ genome editing. So because I have been working on natural sequencing for several years, in particular biomimetics, so we use the whole genome sequencing. It's not a GAD-seq or any computer program. We use whole genome deep sequencing to detect any potential off-target. So this is the method. It's from the parent, get egg or sperm, and then fertilize, inject Cas9 and get RNA. Then we develop for a few days, get sometimes we get a single cell, sometimes we take the whole cell, the whole embryo for genome application, then do deep sequencing for whole genome. At the same time, we sequence the parent genome as a control reference. And we develop biomimetics pipeline to filter out by parent genome, by mismatch similarity to get RNA, and also by quantities. And then we could pull out a list of putative off-targets. And then we use the Sanger sequencing to validate one by one to see if they're real off-target or not. So we are talking about the CCR5 gene. We just want to lock it out. It's easy for HIV infection purpose. And we select the get RNA with one of them with highest efficiency in the cell line. And then we, I want to mention we also have a parent genome, so we could say, for example, in this case, the embryo has a deletion here, while in the father-mother, there's no such a deletion. There's this maybe this is off-target. But for this case, the embryo has deletion, but one of the parents also have this deletion. There is not off-target. It's inherited from the parents. And we are looking for the sites that show some similarity to the get RNA. We see mismatch like 6. That's pretty much most people think it's possible to have off-target. So if it's a 6 mismatch for get RNA, we did the computer simulation to see how many sites in the whole genome differ 6 base pair to the get RNA. It's up to 8,000 genomic sites in the whole genome. And here's we get for the genome coverage. For example, this monkey. For the monkey, the father-mother added the embryo and also control the embryos. And for each sample, we sequence the 40 gig base pair, which is 15x coverage. So for whole genome coverage, if we see at least one read covered, it's about 80 to 90%. But we defined effective coverage, which means for the position, it must be covered by a father, mother, and embryo with sufficient depth of 3 samples. So for the monkey, on average, we get a 45% effective coverage. For mouse, we did 23, and humans, 31%. This number could increase. We increase sequencing depth. Okay, so here's our result. It's for the mouse, monkey, and the human. Again, that's the human. For one of our embryo, we could detect more than 50,000 deletions among the whole genome. And then we filtered by the parent genome. We could reduce to 30,000 for deletion and also insertion. And then we remove some low quality index. And the last filter we use is the mismatch with 6 and also the pan sequence. and we get a number, the 4 or 1. Just that's true for both monkey and the mouse. We can get sometimes get a 0, sometimes get 1 or 2. So there's not a few putative off-target detected by this method. Then we will pull out this off-target, and we do single sequencing to validate it. And here's this is the monkey result. We actually extended the mismatch to 7 mismatch. So we get more putative off-targets. Then by one, we with parents and also the embryo do single sequencing. If this off-target was observed in the parents or controlled embryos, and we see this is not real. And the result is none of the off-target are real. They are all false positive. We can always see it in the parents or in the control embryos. So, and we also did top 30 predicted off-targets by Fenzhang's computer program, and they are not present in the embryo. So put the number together. See if the should get on the mismatch from 1 to 7. We could the total size in the genome, whole genome, in the case of the monkey, it's from 0 to 50. In total, 60,000 genomic sites. And if the whole genome deep sequencing, we could detect some putative off-target. It's in total 19 sites. And so we validated one by one by single sequencing, and no off-target is confirmed. But I want to mention that it's only for 45% of effective genome coverage. So this is for, I would say, good get-RNA design. But what happens if a bad get-RNA? So we take the get-RNA from the guide-seq paper, targeting the VEGFRA, and it's well documented off-target thing here. So we inject this get-RNA to human embryo, and we have 8 human embryo, and we just screen the off-targets. So for every single embryo, human embryo, we could detect at least 1 off-target. So the embryo off-target is confirmed for some bad get-RNA. It's kind of positive control. Okay, so that's the first part about the off-target. The second, and look at the mosaicism. So do this is have an embryo, then we get every single cell from the embryo to single cell whole genome application, then sequencing them. And we can determine the genotype for every single cell, whether it's, the editing is happening or not. So so we have done this for 16 batches of human embryo. In total, we did over 300 human embryo. Mostly are 3 p.m., some are 2 p.m. And we tried multiple methods. And the efficiency is getting better and better. So currently, put all the data together, the average editing efficiency is 32. And 32% embryo has shown some editing. And for mouse, it's better. It's 90%. For monkey, it's 70. For humans, only 32, maybe because the embryo is not healthy embryo. And the Cas9 protein is better than mRNA. And 2P and 3P are not similar in the efficiency. And then the India pattern, we saw that the monkey and the human share some India pattern. And they are most common as just one insertion, and there also one insertion here. Because the mouse and the monkey, we use the exact same get-RNA targeting the same place, so the patterns are similar. And the top 3 patterns are auto-free, which is good, lock out the CCL5. And then the mosaicism, for the embryo, we get for example, get 1 or 6 cells from the embryo, because this embryo usually developed to 4 or 6 cell stage, then stop. This is not a healthy embryo. And for every single embryo, we determine the wild type, heterozygous, or homozygous lockout. And the result is the majority, almost 70% of embryos are mosaic. And it's very common. And we tried to solve this problem by first the timing injection. We could inject the CRISPR-Cas9 like 6 hours after ICSI, or together with the sperm. And injecting with the sperm is more efficient. And we also see, this is the monkey result. So the monkey can develop to blood cell rate. It's healthy. So we evaluate how many of this embryo could develop to the blood cell rate stage. That means to the healthy ones. And the CRISPR-Cas9 seems doesn't affect much the embryo development. And the earlier the injection, the better to reduce the mosaicism. The second one is the double injection. So we could inject at one cell stage and then develop 2 cells with deal again. So double injection, it's better. I could have higher efficiency and it's more homozygous lockout. All right, so just put the data together. It's, so off targets could be minimized by carefully designing get RNA, and whole genome sequence is a very valuable tool to deal off targets. And the majority of embryo mosaic. And the very last one is, I want to put this, it's a genome editing for human, a germline, it's going to happen in the future or near future. So I want to remind everyone, we should do this kind of slow and with caution. It's, because a single case of failure may kill the entire field, just like this case, the biotech death of just canceling for the gene therapy. Okay, so with that, thank you for your attention. Yeah.
So you suggested that injecting with the sperm is at the same time as the sperm in ICSI is the best way to go. Are you modifying both genome? Like when you say there's 85%, is that both the maternal and paternal, or only the maternal genome is edited?
We're not distinguishing maternal or paternal gene. Yeah.
Well, I guess the question is more, are you getting biallelic targeting? Is that 85% having one or both genes modified, both alleles?
Oh, so that, yeah, the homozygous or heterozygous lockout. That's yeah, that's the. I think it's in this case, yes. So most of them are mosaic, and most are just a single copy lockout. If it's a homozygous lockout, then it's the few embryo. It's every cell has. Oh, I think it's for every single cell we could quantify whether it's a double lockout or just one strand.
I have a question too.
Yeah.
So Kathy Jiankui yesterday talked about also editing human embryos.
Yeah.
So can you comment on the differences or similarities of your methods?
Well, it's a very different study with cancer. So we're looking at the off-targeting and the cancers developmental biology. I don't know how to comment on that.
So is that table meaning that you're injecting 10 different guide RNAs into one cell?
No, it's a 10 embryo. For every embryo, it develops multiple cell stage.
Oh,
then I get the cell. For example, this one, all these 3 cell stage, was then take every single cell and then analyze it.
Great. Let's then. Oh, one more question.
So in your first analysis, did you also look at the SNPs or just indels?
Currently, we only look at the insertion and the deletions. We're not looking at the SNPs at the moment.
I see. Thank you.
Great. Thank you for a great talk.